following is a conversation with Dr. Ran Blekhman, Associate Professor at the University of Chicago. Ran is a geneticist by training, but is applying his genomics background to the study of the microbiome. In this episode, we delve deep into the concept of the microbiome being our second genome. We spoke about how our second genome has vastly more genes than our human genome, and how potentially some of these genes might be heritable, i.e. they come from our parents. I reached out to Dr. Blackman a couple of months ago after he published a paper in Cell with colleagues where they analysed a massive data set of over 168,000 samples, and they've drawn some really interesting insights and data from this analysis. So that was a feature and focus of the conversation that we had on Inside Matters. We also explored some of the emerging research that came out of his laboratory, all around the interaction between the genome and the microbiome, but also we talked about some of the early life microbiome and breastfeeding and how that might impact on the microbiome of people as they develop through being a newborn and into infancy. So really fascinating conversation. It was a little bit more technical, I think, than some of the other conversations we had on Inside Matters, so it might be better suited to people who've got some scientific training. But even if you don't, I still think you'll get something from the episode. Podcast is going great guns. We see an increase in the number of listeners on a month-to-month, quarter-to-quarter basis, which is exciting. I feel like it's really starting to take off and hit kind of escape velocity. If you haven't yet liked and subscribed, please do. That's the best way to support the podcast and keep it free for you as a listener. This is Inside Matters. My name is Dr. James McIlroy. I hope you enjoy it. Well, Ran, thank you very much for joining us here on Inside Matters. Pleased to have you.
Of course, I'm happy to be here. Yeah, thank you for having me.
So why don't we start then with a little bit of an exploration into the second genome? And based on your background as a geneticist, why don't you walk the listener through our genome, but then this second genome? What are the key differences? How do they intersect? And why are they important?
Yeah, of course. So the human genome is of huge importance and interest to scientists everywhere, and it has been for a very long time, right? We always knew that our genome controls susceptibility to disease, controls any traits or phenotypes that we have. It's a really big goal for research, biomedical research in general, is to find the genes that control different traits and different diseases in order to then eventually maybe be able to cure diseases or understand the mechanism of diseases, right? And something kind of new that's had been emerging in the last maybe a few decades is that we have like what you just called our second genome, which I assume you mean the microbiome, right?
I do, yeah.
Which is the genomes of all the microbes that are found in our gut and everywhere else in our body. And we're finding they're also relevant for disease and other traits and could be important to look at. And and what my lab is really interested in is this kind of intersection between our genome and the microbiome, and then seeing how they might be interacting, right? Can our own genes maybe determine which microbes we have in and on our body? Can the microbes in turn, affect our genes by maybe changing their genetic regulation? And eventually the goal is then look at this interaction between our genes and our microbes and see if this interactions can affect all kinds of disease and other relevant traits.
Thank you. So before we delve into the details, could you just maybe take a step back and talk a little bit about maybe how many different genes there are within the microbiome versus the human genome, and what's exciting about the human microbiome from a genetic point of view? Maybe it's easier to modify, these kinds of things.
Of course. Yeah. So in our human genome, we have about 20,000 genes. It's a small number compared to what we have about microbiome, right? In the microbiome, if we think about all the microbial genomes and all their genes that are different, right? You can have many, many more orders of magnitude more, maybe millions of different genes in our microbiome. So if you think about the potential different functions they can do, metabolic functions, it's more than what our own genome can do. On the other hand, our own genome is directly obviously important for our physiology, right? Because our genes basically control everything's happening in our body. So this is a very important area of research, right? The idea that, there could be an interaction or some kind of an association between our genome and our microbes. For example, if could be a disease, and if you have a certain genetic variant, you might be susceptible for this disease, and the presence of a microbe or maybe a gene in the microbiome can increase susceptibility and maybe lead to the disease actually manifesting. So that is kind of the idea, the basic idea behind our research. Yeah.
If there's millions of genes, how much do we understand about them and their functions? Like, is it well known, or is it still an area that's really in its infancy in terms of understanding?
Yeah, a lot of it is known, right? A lot of specific microbes have been studied pretty deeply. On the other hand, there's a lot we know very little about. And we know from our studies, when we do metagenomic sequencing, we sequence the microbiome, we find a bunch of genes that just seems to be well characterized, and we know what's going on there, we know what they're doing. On the other hand, lots of genes where it's unknown. It's probably a gene, it looks like a gene, but we have no idea what it's doing. We can maybe guess sometimes based on similarity to other genes, right? But in many cases, it's unknown. So there's a lot of unknown, and I would say the amount, like how much is unknown, kind of depends on which microbiome you're looking at, which kind of leads to this recent study that we had. We're trying to kind of look at microbiomes across the globe.
Yeah, and we will definitely talk about. I want to build up to that paper because it's new, and that's of course why I reached out to you, because I read it and I thought this is extremely interesting. But just going back then to the genetic piece for a second again, is it that is there like an infinite number of genes because the microbiome is unique as like our fingerprint, and every single person has a slightly different genetic code within their microbiome? Or is it kind of like well established that there's this fixed number, which is like 2 million and no?
No, I don't. This is a really good question. I don't think it's a fixed number. And I think because there's such huge diversity in the microbiomes, you would see, just if you think about the number of taxa, you would see individuals that have a lot more versus a lot less. It could depend on where you're from, it could depend on your diet, it could depend on your genetics, right? It could depend on a lot of, depend if you just taken antibiotics, for example, right? Reducing the diversity of the microbiome. And less diverse microbiomes, I would say, more likely have much fewer genes in their genome. Just you have fewer taxa, fewer genomes, fewer genetic diversity. Yeah.
Gotcha. And are there any like quite cool and neat trends? So like, for example, in Japan, are there lots of genes that help you metabolize seaweed? If you're in Scotland, maybe there's some genes for whiskey, I don't know, or something like that. But you see what I'm saying? Like, are there adaptations, are there sort of genetic adaptations depending on where you, maybe that was a bad example, but I'm based in Scotland, so.
No, I have, actually, I haven't heard about the microbial genes that metabolize whiskey. That could be interesting to look into. But there are kind of known patterns that are correlated with diet. I think one of the more well-known ones is Prevotella and Bacteroides. So Prevotella is the one microbe is kind of known to have higher abundance in, say, non-westernized countries. It's more kind of associated with diets that's kind of higher in fiber. On the other hand, you have the Bacteroides, which is kind of much more abundant in, for example, the US and Europe, and this kind of more associated with the Western diet, high-fat diet, processed foods. Yeah.
Got you. So how much do we understand about the interaction between the human genome and the microbiome and vice versa? Like what is the current like understanding of this interaction? Because if there's 2 million, maybe more genes, that must be a very complicated set of interactions, right? Because there's so many different potential functions. So what do we understand about this very intricate and dynamic relationship?
Yeah, this is a great question. I would say we're starting to understand it. So this is kind of a new area of research, and we're kind of getting there. pretty slowly. It takes time for this research, the literature to build. But I would say what we're finding out is, I would say, first of all, we haven't even looked at, so far, very little, we haven't looked at the actual genes in the microbial genome. We more kind of the beginning where we're just trying to understand what is the effect of human genetics or my own genes on the overall composition of the microbiome. So which taxa are there, maybe the abundance of certain taxa. I don't think we've gotten yet to certain genes or pathways or functions in the microbiome yet. We're not there yet. So a lot of the studies that have been done so far on the kind of association between human genetics and the microbiome are more kind of trying to understand whether this microbiome in general, how much of it is genetic, how much of it is controlled by genetics versus how much of it is environmental. And the approaches people have used to do this come from human genetics background. I mean, you can probably, you probably know that in human genetics, understanding kind of the genetic basis of different traits has been something people have been doing for hundreds of years. So, nature versus nurture. How much of this trait is actually genetic versus how much of it is environmental, right? And people have been doing this looking at traits like, height, weight, and so on, right? Yep. And what we've been doing and others have been doing is basically looking at the microbiome as sort of a trait of the host, kind of like height, right? And then using these kind of standard approaches from population genetics, human genetics, to try to quantify how much of this trait, microbiome trait, is genetic versus environmental. That was kind of the first step in this kind of field.
And just to unpack that then a little bit further, how much of it is inherited, so to speak, or heritable, and how much of it is kind of the nurture element? Because one of the fascinating things about the microbiome, having hosted this podcast and spoken to lots of experts like yourself, is perhaps unlike the human genome, it's inherently modifiable. As in, you change your diet and it changes quickly, whereas the genome is different. You need to kind of engineer it.
Right.
So where have we got to then in the field about, is it, your parents, or is it more you and what you do?
Yeah, and you're touching on several interesting points there. I think one of them is the concept of heritability, which you mentioned, which is basically the kind of the population genetic statistic or name that we use to assess how much of a trait is genetic versus not genetic. So we call it heritability, and we say things, for example, like height is about maybe 70 to 80% heritable, which means it's genetically controlled. Yeah. And other traits, you usually from there you kind of go down to less and less heritable traits. You have like personality traits that are much less heritable. And what we're finding with the microbiome, and this is based on kind of research from my lab, other labs, is that the heritability of the microbiome is not super high, meaning it doesn't reach the high heritability of, traits like height, for example, or other like body measure traits, but or it doesn't reach like heritability levels of certain diseases, which are heritable, are genetic.
That's interesting.
But it is higher than 0, and some of the estimates we're seeing are between 10 to 20 to 30%. Now that's very dynamic. It changes person to person. It changes based on which taxon you're looking at, right? Some parts of the microbiome are more heritable, some parts are less heritable. It kind of also depends on all kinds of technical factors we don't need to get into, but like how, for example, how you define your microbial traits. What do you use as the microbiome trait, right? So, but so in general, it is
quite interesting. I am quite interested in what those could be. So, is it just like, is your microbiome diverse or not diverse, or like what are some of the traits? Because I think that is interesting, like the technicalities. Let's go a little bit deeper.
I'm happy to talk more about it. So diversity is one kind of obvious thing, like you said. So you can look at the diversity of the microbiome as a quantifiable trait that you can look at, and then you can see how heritable is this trait, the diversity of the microbiome. And we're seeing this is one of the more heritable microbial traits in general. Like diversity is pretty heritable, right? On the other hand, then you can break this down a little bit and look at certain taxa and look at their presence or absence, right? Whether a taxon is present or not as your trait. It's kind of a binary trait, right? Or you can look at the abundance of that of a certain taxon as your trait. In this case, it's kind of how much you have of a taxon, right?
Interesting.
Yeah, you can then define more complicated traits, like how much a taxon is changing over time. That you would need to have some kind of time course sort of data, the microbiome data to look at that. Yeah, the challenges we look at the heritability of the microbiome are pretty big, right? As opposed to something like height that doesn't really change once you reach adulthood, right? The microbiome changes all the time. It's really dynamic. It changes day to day. So to actually quantify the heritability, you need more kind of longitudinal data. And this is one study we had from the lab in collaboration with Beth Archie from Notre Dame, where we looked at a population of wild baboons in Amboseli in Africa, where we actually had very long-term longitudinal data. So dozens of samples, almost sometimes more than 100 samples for each individual, covering several years of life. And we have the complete pedigree of this population of baboons. So we can know completely the relatedness, the genetic relatedness between each pair of individuals. And that kind of allowed us to control for this longitudinal dynamic of the microbiome, while at the same time try to quantify all kinds of those heritability factors. And what we're finding from that study is that the heritability of the microbiome is relatively low, regardless of which factor you're looking at. It kind of ranges between 10% to 20 to sometimes 25 or 30% of the rare, very heritable microbiome features, meaning it's more environmentally controlled than genetic.
Gotcha.
The microbiome. But we're also finding that almost all the taxa that we looked at are heritable. So almost every part of the microbiome is heritable, is significantly heritable at some level. So it seems like there's a genetic control on every part of the microbiome, but that genetic control is not huge. It doesn't play a huge part in controlling the composition.
That's fascinating. How good is Baboon as a model system?
It's actually pretty good. Obviously it's not a human, but there were some studies that showed that the baboon gut microbiome is the most similar to humans from all other primates. So it's actually a pretty good model system. Yeah, it's actually a pretty good model system to look at the human gut microbiome. Yeah.
Because I was at a conference last year and there was a big debate about, what is the best model system microbiome, and Baboon didn't come up. I think there was lots of other suggestions. So it's cool to hear that. So next time I hear that debate, I'll say I think the Baboon might be but
specifically for that system, this is an amazing data set because we had this longitudinal data set covering years and years of life, sometimes the entire lifespan of some individuals. And we had a very good observational data on their diet and environmental interaction. So we can control for individuals who are interacting with each other more. We could control for exactly their specific dietary components. So we could do all that and actually control for all these factors that could bias other, standard kind of heritability analysis. So this was the advantage of that specific system.
Gotcha. No, thank you very much for that. So I think what we're saying is it seems to be heritable, but the extent of the heritability is sort of still being understood. And
yes,
you're trying to figure out the dynamics. Okay, that's fascinating. Thank you. So is there something to do with maybe the co-evolution of us and our microbes that could be important here? I guess it's maybe not an accident that they're inside of us. So how much do we understand about these functions maybe helping us in terms of our genome?
This is a really interesting area of research. And I think there are more and more, research labs are interested in understanding this kind of coevolution dynamics between the host and the microbiome. Personally, I would be very kind of careful of using the word coevolution because that means there's some kind of a genetic adaptation in both the host genome and the microbial genomes that allowed each other to allow them to work together, to adapt to each other. And this is something that's really hard to show. You have good evidence to show you actually have coevolution. I would say something we do have is another word we use is co-diversification, which means certain groups of hosts have certain groups of microbes associated with them. So certain populations of humans seem to have more specific populations of microbes. And if you plot the tree, the phylogenetic tree of the host and the phylogenetic tree of the microbes with each host, you would see them kind of correlated strongly with each other. And that would mean basically there's some kind of an association between hosts. Certain groups of phylogenetic groups of hosts have certain phylogenetically cohesive groups of microbes, right? And that could be because these hosts eat a more specific diet that attracts these microbes. So it doesn't necessarily mean there's a genetic adaptation to each other. It could be like both of them are environmentally more kind of adapted,
Gotcha.
you know?
Gotcha. So if they are heritable, how does it happen? Is this where the kind of maternal piece comes in? Is it you share living environments, or how? Because we're kind of taught we're born almost sterile, right? So
yeah.
What's the mechanism then of the heritability?
That's a good question. So first of all, just to make sure to clarify, is that heritability means it's genetically controlled, right? If something is heritable, let's say I say this microbe is 20% heritable, it means that 20% of the variation in this microbe abundance, for example, across the population of individuals we're looking at, 20% of that variation is due to genetic variation in the host. So that doesn't necessarily indicate there is some transmission of that microbe from mother to offspring. It just means there's some correlation with a certain genetic allele. So for example, it could be if your mother or my mother transmitted a certain genetic allele to me that helps digest a certain fiber, right? So that means that microbe would have more food. So you have a higher abundance of the microbe. So that could be just an example. One of the most heritable, I can give you one example of like the most heritable gene, like host genes associated with the microbe in humans is the lactase enzyme. So genetic variation in the lactase enzyme is very correlated with the abundance of bifidobacterium in the gut. So bifidobacterium is a microbe that really likes lactose, and some studies actually show it prefers lactose over glucose and really grows well on lactose. And in humans, lactase and the enzymes that basically digest lactose.
Yeah, gotcha.
So there could be a few, I know there are a few labs who kind of try to understand the mechanism here, but there could be different ways to understand it. One way could be certain genetic variants in and around the lactase enzyme, the lactase gene, basically determines whether someone is lactose intolerant or not. Wow. And that determines your diet, which in turn determines whether that bifidobacterium would have food, basically.
Yeah. Wow.
That's one way to think about it. Yeah.
So could it be that the lactose intolerance is microbially driven?
So this is a good question, and this is a kind of active area of research for a few labs, because it's kind of the lactose intolerance is something has been studied in human genetics very extensively, and because this is one of the strongest signatures of selection you have in the human genome. It is one of those things that I teach when I teach human evolutionary genetics to the undergrads here at University of Chicago. Is one of the most interesting kind of best examples we can give of human evolution, human adaptation. That's pretty recent, because basically being able to digest lactose into adulthood has given people a huge advantage, especially in times of famine and milk is also a very good nutritional source. There's a lot of things there that are really helpful, right? So being able to digest it give people a lot of advantage. So you see a strong selection in that area of the genome around it. The gene is called the LCT gene. In humans, mostly in like European populations, there were some studies that have shown similar selection happening in other populations at the same time. Also, probably, giving this kind of advantage of being able to digest lactose, right? But there seems to be some kind of some cases where some human populations don't have this allele, but still able to digest lactose, meaning they can drink milk and don't have the, the phenotype you get, right, if you're intolerant. So there's really interesting active research going on about trying to understand maybe the microbes are actually helping their human host digest the lactose, and that could be a really interesting idea. But I haven't seen any conclusive evidence that it is happening yet. But I think it's a really interesting question.
That would be fascinating. It would be really cool if there was some microbial driven aspect to that, because that would really add to the sort of broader story which you were talking about there. So, so as you're looking forwards then, and I want to get to the Cell paper in a bit too, but how do you see our understanding of the interaction between the human genome and the second genome, the microbiome, sort of translating into, diagnostics, therapeutics? How could you see that kind of coming together as we understand the interaction in a deeper sense?
Yeah, I think we're just getting started. So we're still in the early stages of trying to understand the heritability in general and trying to understand how much of the microbiome is genetically controlled versus not. The way I see it is actually a very promising area, because it could help us understand if certain genetic predisposition to disease can actually be, or at least our understanding of why some genetic variants lead to disease versus not, can be because of this environmental factor, the microbiome, right? We know that you need a certain gene, but you also need like a certain diet together to lead to, for example, I don't know, type 2 diabetes, right? So could there be a microbiome component that also changes the susceptibility, where your genes and microbes kind of work together? So you can think this might be some sort of a diagnostic, right? So if you have this gene, but you also have this microbe, you are at a higher risk, which would mean you could have a different kind of recommendation, like the clinical recommendation of what to do to lead to a better outcome based on your microbiome and genome information combined, right? There are more specific studies now looking at specific diseases like inflammatory bowel disease, where there's certain genetic factors and certain microbial factors that might be working together, but this is kind of more specific stories there.
Gotcha. And maybe it's a good time then to segue into some of the population scale insights that you've been working on. So maybe we can just really unpack the paper then. I think 168,000 samples or so this is big. I think it'd be really cool to hear about how it came together, maybe global patterns that are emerging, perhaps maybe some of the Western bias that we see in terms of the data, and just the key takeaways and what you're excited about, because it got quite a lot of traction. And of course, I reached out to you as soon as I read it, because I wanted to talk about it on this podcast. So it'd be great to hear more about that, and congratulations on the publication.
Of course, thank you. Yeah, and I have to say that the work was done, almost all of it, by Rich Abdul and Samantha Graham, were the first authors on this paper. And also we had some amazing collaborators that helped us pull this together. And like you said, it's a major effort. It's a huge data set.
Big data set.
I think this all started, if I think of kind of what motivated us to do that, with Rich, when he started his PhD in the lab, which was years ago. And his question was actually still related to human genetics and the microbiome. And he was doing some, and I don't even remember what the analysis was, but he was trying to find some correlation between human genes and microbes. And he came to the realization that you need publicly available data to kind of get some kind of a baseline. So from there, you kind of move to like, okay, let's start collect some relevant public data sets to use for this other project. And as you kind of went on with looking at publicly available data, you realized you can actually pretty easily collect lots of publicly available data. Easily is an understatement, but you can collect publicly available data and kind of integrate them and get some and learn something about overall data, overall the microbiome, what's going on in the world, right? And he kind of developed this whole system for downloading and integrating raw microbiome data into this one big data set. And the more it went on, he realized, okay, this is actually very doable. And this is actually really exciting and interesting as a biological question, right? And as a resource for the community. We have this huge data set of everything combined. It is something that's could be helpful for a lot of other microbiome researchers, like to get a baseline of whatever they're studying, right?
And did he have to engineer the data so it was all similar format? Like how did he manage to do this? Like when you talk about public data set, what is it? It's like spreadsheet code. How does it look? Just for the listener, how does he actually do that? Because it's probably quite a difficult thing to do.
It's definitely not easy. So public data. So we're talking about specific type of microbiome data that's called 16S sequencing, and this is a sequencing of one region, a few hundred base region that's found in all bacteria. And this kind of an approach is used to understand the basically what species of the microbiome are there in their abundances. You can't really look at any genes in the microbiome, but because the sequence is very different between different microbes, you can kind of, for each sequence you're getting from the sequencing study, you can understand which species it came from. So we realized if you download all those sequences for each study, you can download the actual sequences data, and then you can use kind of a standard pipeline for processing it. And the pipeline is, once you kind of you run it in multiple studies, you can integrate more and more studies into this one big integrated table. You can even think about it as kind of an Excel spreadsheet where you have all the samples that were ever collected and in the rows, and the columns are all the taxa. And each cell would have the abundance of that taxon in that sample. Okay. So going from the sequence data, which is, a bunch of ACs, Ts, and Gs that represent the microbiome in each sample, to that spreadsheet is kind of the more complicated process, but Rich found a way to standardize that so that every study can go through the same pipeline and kind of add to the same big spreadsheet.
Hey, cool.
That had all the microbes, yeah. And that was the outcome of that study, is this big spreadsheet that is basically representing all the human gut microbiome data, specifically 16S microbiome data that was publicly available. Yeah.
Gotcha. Okay, this is cool. And in terms of the kind of spread of data, where was it more clustered to? Like, what exists so far, and how did he kind of scrape that and pull it together?
Yeah, so the challenges, I would say, this is all coming from several specific databases, right? And the reason there are a small number of big databases that hold all this data is that researchers, when they publish a paper about their data set, they have to upload it to that same database, which is, this is kind of a requirement from NIH, for example. If you get a grant from NIH and you publish anything, you're kind of required to also upload the data. A lot of journals, if you publish a paper, they require you to actually make the data shareable, which makes sense, right? Because you want all the research to be reproducible. You want it to be usable by the community, right? So having all the data available is great. But the problem is that not a lot of people were using it. It was all there. There were terabytes and terabytes of data available on these big databases, which, one of these databases, for example, is at the NIH, and they're spending lots of money, to maintain it and keep it stable and have the website and have like an interface that everyone can download whatever they want. And based on what we know, some people use some of these data sets, but usually people use one of just the biggest data sets. So you take the biggest data set and reuse it. And there are some data sets there that are reused a lot, but most of the data is not used. And this is because a lot of the microbiome studies are small studies. They're studied with like 50 individuals, 100 individuals, 100 samples, and they're uploaded to this database, and then they're not reused at all. Because it's a small data set that's probably not relevant for anything specific that someone else would want to do. But what we found is by taking all these small studies together and integrating them, you're actually getting a really great kind of eagle's eye view of the world's microbiome.
Okay, fascinating. So he was pulling together data sets that had been well described, well used before, but also some of the more smaller ones, and then just pushing them together. So did he have did you guys, did the team, did he have had enough hypotheses going into this, or was he just doing this cool thing, which was, we're going to mix all these together and see what comes out?
Yeah. I mean, we definitely had some hypotheses based on what's known about differences you would expect in the microbiome between world regions, right? Like we knew, for example, that Bacteroides, you already mentioned, probably you would see it a lot more in Europe and North America, less in other regions. And it was found in other studies that looked at differences between continents and between populations, smaller studies than this one, but you would see this pattern pretty clearly. And we were able to find that pattern, kind of reaffirm that this is there. But overall, we didn't have a huge hypothesis, right? Our goal was to make this as kind of a resource for everyone, but then also try to see what we can learn from it. And so this is one of those studies where we just generate this very big data set, and we thought about things we can test, but in some cases we didn't actually have a hypothesis of like what we expect to see. It was all like, okay, let's see how much we know about the microbiome in different world regions, and can that tell us kind of what we're missing, for example, in regions that are undersampled.
Gotcha. So I guess this brings us to what you learned and what kind of we found and what was published. So why don't you walk us through some of the exciting findings and results and key takeaways?
Yeah. So the one of the biggest things that we found, which was kind of known because we already published about this a couple of years ago, we published a paper that did not have any microbiome data, but just characterized the samples that we're seeing, where they're from. So where each of these studies are from. And what we found is basically almost all the microbiome studies, almost all the microbiome samples are coming from Europe and North America.
Yeah.
A huge proportion, much higher than the proportion of the actual population in these countries compared to the rest of the world, right? And there were some regions that are much less well sampled, right? And so this is something that we kind of knew beforehand. What we could do with this data set that actually integrated all the microbiome data itself, we could actually see what are the taxa, or not the taxa, but like how much are we actually missing? And one of our main findings was that in places like Europe and North America, we're kind of not detecting any new taxa anymore. If we want to detect more microbial novelty, new things that we haven't seen before, you have to sequence a lot. Many samples, you got to go very deep sequencing.
Go deeper.
Yeah. You have to do a lot more sampling. But on the other hand, in places like Africa, South America, we're actually really not even close to saturation point. And even with one sample, a couple of samples, we're getting a lot more for our money, basically. If we're interested in microbial novelty, we're interested in learning about what microbes are there. So that's kind of one of the interesting findings we had.
That is interesting.
Yeah, one of the more important things that we found from this study.
And do the microbiomes, I think I mentioned this earlier about the genetic element, do they look the same? Like, can you tell where someone lives based on their microbiome composition? Is that possible?
Yeah, so this was our second very interesting result, is that there were actually pretty big differences in our microbiome across the world. So you could see very clearly big differences between big world regions, between continents, even between countries. You could see big differences. And one of the things we did in the paper is actually try to train a deep learning model to, based on a given microbiome sample, can you tell me where this host is from, which country this person is from? And it could do it with pretty high accuracy. So we could actually determine, given a microbiome, a gut microbiome sample, give you a pretty accurate estimation of where in the world this is coming from. So the correlation is pretty strong, and a pretty, I think, exciting idea of, like, just in general, knowing that the microbiome is different. Especially, it's important, I think, for any kind of medical purpose. You're trying to understand what is a healthy microbiome, what is the disease microbiome. The baseline is going to be different based on the country you're in, right?
Well, I'm really glad you mentioned that. One of the questions, I think probably the question I asked the most on this podcast, because it's a fun one to ask, is what is a healthy microbiome? It should be easy to tell, right? But no, I know it's more complicated. So maybe you could give your take on this question. And I've not heard it pitched like you just did around, actually, maybe even depends where you're living to get that baseline. So, how would you respond when someone asks you, Dr. Blekhman, what's a healthy microbiome?
Yes, I should preface by saying that I'm not a clinician. I'm not a medical doctor. I'm a PhD, so I can hypothesize based on what we're seeing in this data. But another important caveat to mention is that we didn't actually have health information for this data set.
Huh.
Right now we're processing the metadata that we have for all these samples to try to figure out, are some of these coming from certain disease categories, some are healthy, and so on. So this would be a very interesting kind of follow-up analysis that we're going to do. But at this point, we just had the geographic information. That's kind of the only thing we had. We had some other technical information we could kind of infer from the data, but in general, it's just like the country level, regional information. But to try and answer your question, even in just like the US, I don't think there is an answer to what is a healthy microbiome. I mean, I'm sure some people will tell you, oh, higher abundance of this is more healthy in general, higher diversity is more healthy in general.
Yep.
But I think there's no answer of like, this is healthy versus this is not. And I think the contribution of our study is that there's also population variation where what is a healthy microbiome should maybe depend on where this person lives or where they're from, right? So it's it doesn't help answering the question, even complicates answering the yes.
It's even more complicated. Great. Oh my God, that's funny. So I guess, to be honest with you, the fact that it's possible to pinpoint perhaps which country someone's from is in some respects surprising, in some respects it's not. I think before hearing about the results and reading the paper and so on, I would have said, I think you're probably going to be able to tell the difference between a kind of Western microbiome and then maybe an Eastern microbiome, and the difference between a vegan and a meat eater or something like that. But the fact that it's maybe even country specific, why is that? Is it just this, it seems kind of crazy actually that it's country specific, and it somehow just consistently represents the country independent of ethnicity, genetics, other biological factors. So maybe you can, am I misinterpreting, or is that correct? It's sort of...
I think I think this is a very good question. I would say that I think a lot of those country specific differences could be because of genetics, diet, environment, and so on, because we don't have that information in our data. So everything that really seems to be country related could be driven by a lot of these other factors.
Yeah, fair.
That, genetics makes sense. Diet definitely makes sense. Environment would be different environment. Each country has different medication procedures, antibiotics use, lots and lots of things that could be all grouped into this country label, right? But I also have to say that the country signature in some cases was strong, in some cases wasn't that strong, right? It could be, small percent differences between countries that we're able to see because we have such a huge data set. So I was saying, do you significantly say which country it's from? We have such a huge data set that allows us to do that, but it could be that differences are not always very big.
Gotcha.
Like you said, we know there are very big differences between, let's say, the US and, East Asia, right? But we found huge differences, right? But from some other countries, it could be pretty small differences we're able to see just because we have so much statistical power, analysis.
Gotcha. Because I'm running a drug development company focused on the microbiome, and we just finished phase 2 trials in IBS, and we're planning for global phase 3s, and we're speaking to larger companies that are further down the path to us. And one of the most common questions, particularly from companies based in, say, Japan and South Korea, is, James, your data looks very exciting. This could be transformative for some patients, but your donor program is based in the UK, and therefore your microbes have been derived from donations from someone based in the UK. Is it going to work in Japan or Korea and South Korea? It's one of the questions that we get asked the most, but we haven't done a trial in those countries. So it's kind of difficult. Maybe you could just give a kind of gut reaction, if you don't mind me putting you on the spot. What are your thoughts on if an LBP has been developed in, I'll say, the US? Is it going to work in Japan? Like, are the differences that big? What would be your expert kind of gut take on that one?
My answer is that I don't know. It's a really good question. Yeah. I don't think it has been tried, right? I think your company is one of the leaders in the field, and even you've only looked at this, in the same country. I don't think anyone has tried that. And I'm very interested in seeing what you find in your clinical trials in other countries. I think it makes sense to try to have the donors come from that country, or at least do some kind of a profiling of the microbiome to see that the strains in the LBPs are kind of consistently found in healthy individuals from that country. I think that makes sense.
Yeah. So we think about it very similar, I think, to how you've articulated it. Not to go into any kind of secret sauce or anything like that, but I mean, what we do is we pool together microbiomes from different people, and then you end up with something which is inherently more diverse than any of the individual donations or any individual donor. Mm-hmm. And we think that gives us more shots on goal, more shots on target, more functional redundancy. And then the other thing is, like, the way I explain it to a potential partner and collaborator is, we have more chances of success because there are simply so many microorganisms in our product versus a defined consortia that has maybe 6. We see hundreds in all of our products. We also see these core, consistently present groups, and they are the short chain fatty acid producers, they are the bile acid metabolites. That instills confidence. But the other thing that we say is, look, in contrast to a different product where they've said these are the 6 bugs, mm-hmm. we absolutely could set up a donor program in Japan or South Korea, and we could take a donor pool from there and run it through the same process. So that, right, there's pros and cons. There's pros and cons, and it's really interesting to hear your thoughts, because I think it's going to become more and more of a theme as hopefully we see more and more drugs get to phase 3 and beyond, because that's what we need. Mm-hmm. So, thank you for your insights on that one.
I agree, but I think this is a great approach, because you have a lot of different taxa there, and you basically, whatever sticks might be the helpful, and using donors from that.
Sorry, please go ahead.
Yeah, using donors from that country, or at least doing some kind of an analysis in the product has strains that seem to be represented in healthy individuals from that country. I think makes a lot of sense to me.
Yeah. Well, we did map some of our bugs versus some of the data in your in your publications. we did have a look at that, and indeed, they are similar. But maybe you could have off the podcast. Maybe we could have a chat about that, because we might be able to do something quite neat there, actually. Anyways, back to your research. Sorry, I don't want to talk about. We're here to talk about your research. So. What else kind of surprised you then that was kind of interesting as you pulled together this huge data set, and then maybe we can talk about the kind of next steps, because it sounds like there's more to come, which is cool, like the metadata.
Yeah, I think one thing that kind of surprised me was kind of on the more technical side, is that there are technical factors in how samples are processed and and preserved that had a big effect on the composition of the microbiome. So one of the things we looked at, for example, because this is 16S sequencing, we could see which region of the 16S was targeted. There's several ways to do 16S sequencing because targets several of variable regions that are found in the microbial genome. So which specific region, we can actually know. We don't need metadata to know this because this is represented the actual sequence, right? We look at the sequence and we know which variable region it comes from. And the variable region, which variable region was sequenced, had a big effect on the microbiome. Another thing that we found, also really technical factors that was very interesting, and I think that's related to your question about the metadata, is that one thing that we did want to look at is specifically how the sample was collected. There are several ways you collect the microbiome sample to preserve the sample. And there are several ways to process the sample to go from a fecal sample to microbiome sample, to like a next generation sequencing library, right? And to extract the DNA from a sample, right? And we were actually able to do, and this is kind of leads to the next question of how do we get metadata. This was done by kind of reading through the papers and using kind of a computational approach to go through the text of the paper and find the kind of the text that matches which kit was used to extract DNA from the sample. And we found that basically for each study, for each sample, what kit was used, right? And from that, we can see what are the properties of each kit. And what we found is that whether the kit had a bead beating step or not had a huge effect on the microbiome. So bead beating is one of those steps when you process a microbiome sample, you have a lot of microbial cells, you kind of do this step where you kind of try to break down the cells to get the DNA out of the microbial cells, right? And sometimes you have a bead beating step, which is a step where you kind of mix all the cells together to try to break them down. And not just that this had a big effect on the microbiome, there are specific taxa that were just not represented if you didn't do a bead beating step, and vice versa.
So interesting.
And I think that could be related to, some taxa just have thicker cell walls or different cell walls that if you don't have that step, you just don't get DNA from those samples. So this is one of the things that I was really surprised by, how big this effect was. This kind of technical, seems like a technical effect, but there are some taxa that are just going to be missing completely from your data if you don't do that, right?
Wow. Now, how much of the kind of, sorry if this is such a buzzword, AI tools are you using to help with this? Because so much data. Are you using like machine learning approaches? You mentioned deep learning. Maybe you could talk the listener through some of these things, because that's pretty interesting.
Yeah, so in this study, we used a little bit. It depends on kind of what you mean by AI. Some people mean AI, they think about like ChatGPT or large language models, and some we use a lot of deep learning and machine learning in our study, which are kind of established approaches that don't have the buzzword of AI, but sometimes they can't get grouped with AI. But for example, this predictor that I told you, like figuring out accurately predict where a sample came from, that was using deep learning, right? And this approach to kind of find what which kit was used for processing the sample was done with natural language processing, which is another approach. You kind of read through text. We haven't really used any, what I would say, like AI tools, but this is something I'm very excited about. The next step, as we're thinking about, okay, can we get more, let's say, clinical metadata, more information about these samples? Like, are these just samples from individuals who have a certain disease? What disease? Can we get their age, their sex, other information about the sample? Because then we can get a lot more out of this data. Look for differences related to disease, differences related to other interesting factors. And that we're probably going to have to use more advanced sort of deep learning tools to kind of read through papers to find where the metadata is, because it's kind of inconsistent in the database that we're using. People upload their data, but usually don't fill out the metadata labels very consistently. So this is kind of something where it's a little bit more complicated, but we're working on it. This is kind of the next step.
And are these deep learning tools, are they robust enough and good enough for you to use to give you enhanced agency and yield, or are they still kind of early and needing to be refined? How useful are they for what you do?
I think whatever we're using, they're not tools that are used kind of unsupervised. Everything we're using are things that have been tested and used and take a lot of input from us on like what is the right way to do it, what is the right parameters, what do we want to get out of it, and we validate it and make sure it's actually working and so on. We usually spot check, okay, if this tool tells us that all these papers used beat beating and all these papers didn't, we go back and check like 10% of them manually, just to give us kind of an estimate of how accurate it is, just as an example.
Yeah. Gotcha. Okay, so maybe you can then just go a little bit further on the next step. So you're trying to pull in all of this metadata about where the healthy, disease, so on and so on. And what might that lead to then in terms of the kind of future for the combination of such large data sets?
Right. I mean, one thing you can think about, we talked about like what is a healthy microbiome across in different countries, right? Can you say, okay, if you want a healthy microbiome, it looks like this here, but in other parts of the world, all the healthy samples don't have that microbe and have different abundances, right? You can do it. You can think about something like that, or disease signatures, right? The IBD, for example, inflammatory bowel disease in the US looks like this, but in East Asia, for example, it's different. There are other texts that might be represented. So you have a different baseline and a different disease state in each of these countries. Maybe, perhaps, I don't know. We'll find out.
So it could become a biomarker of sorts. This is what it looks like. That's very exciting. That's super exciting.
Yeah, it could be a biomarker that's kind of dependent on, on the specific country, specific population. Yeah.
Excellent. That's awesome. Okay. So with regards to some of the other work that you've been working on, could you maybe just, as we get closer to the end, talk to listeners through some of the other research that you're most excited about within the microbiome space you've been involved in, and we can go from there.
Yeah, we have quite a few very exciting projects. This is just one of them. Another big project that we have with colleagues here at the University of Chicago with Eric Paymer and other great collaborators is looking at electronic health record of patients. And we've now collected for patients here at the University of Chicago Hospital, thousands of patients where we have their full electronic health record. And we have very deep longitudinal data for when they were hospitalized. And this is really exciting because we really, really want to try to figure out what is the association between the microbiome and just this clinical data and different outcomes for these patients. Specifically, some of the outcomes we're really interested in, for example, hospital-acquired infection. This is one of the biggest issues where people coming in and have serious health problems, they can acquire an infection. It could be very serious and deadly, right? So we're trying to see, can we, from the microbiome and EHR, now we're using kind of more advanced AI tools for this, can we get some kind of a risk estimates per patient based on their information in terms of, are they at higher risk? If they are at higher risk, is there certain signatures of the microbiome that are causing this high risk? And maybe there's a way to use LBPs to maybe correct that or lead to better outcomes. So this is one exciting project that we have. Another one that I'm really excited about, really trying to understand this host genetic microbiome associations, but kind of from the other side. So I told you about host genes controlling the microbiome. Here we're really interested in how can the microbiome control host genes or regulate host genes. And this is in collaboration with Francesca Luca, who's in human genetics here. And what we're using is basically cell culture. So human cells, host cells, so it could be epithelial cells. Now we're doing colonic organoids that we're growing in a dish, basically. And we're treating those human cells with live microbiomes that we extract from people, right? The same way we extract for fecal microbiome transplant, extract all the microbiome, treat those host cells. And then you can kind of do a time course and see which genes are responding and how they're responding, and basically you have to learn about the physiological effect of different microbiomes. And we can do this with microbiomes from, let's say, patients and healthy individuals, and also have the organoids from patients and healthy individuals.
Interesting.
So we can kind of understand how differences in the actual host cells affect the response to different microbiomes and maybe affect outcomes too.
Gotcha. So, I mean, do you think, is it possible that our genome is shaping the microbiome all the time, or is it more it's more diet and environment. Like, have you got a view on that? Like,
yeah, that's a good question. So this is a complicated analysis, right? Because you want to understand not just whether you have a genetic effect on the microbiome, but whether you have an interaction of the genetic effect with diet. So you have this allele and you have this diet, then you change the microbiome, right, in certain ways. I have not seen any kind of those kind of interaction effects studies yet, because those would need a very large sample size.
Right, gotcha.
Once you're looking at it, interaction effects. But one of my hopes is that with this kind of culture-based studies, you can actually assess those effects at a patient-by-patient kind of personalized manner, and understand how different microbiomes affect different hosts. And the good thing about this approach, the kind of the strength, is you can kind of change the microbiome whichever way we want. We can like spike in different microbes. We can add certain, drugs, stuff like that. And then we can see how that changes the response of the host.
What about the kind of relative importance of the early life versus the later life? Have you got any thoughts on, as your microbiome shaping just after birth, and as you're developing, are there sort of more important time points to be sampling than others? Like, you got a special interest in anything like that?
Yeah, this is another big project we have in the lab that's led by Kelsey Johnson in the lab, who's basically very interested in the early life microbiome and genetic effects. So would be maternal genetic effects on the microbiome, but especially how does mother's milk affect the baby's microbiome? And we're doing a bunch of really interesting studies, basically kind of mostly almost focusing on the host side by doing genetics, looking at gene expression in milk, looking at HMOs in milk, looking at all kinds of factors that are found in the milk and how they're affecting the baby's microbiome. One thing interesting, probably for listeners of this podcast, is that we're actually looking at the microbiome in the milk, and we're finding a bunch of microbes in the milk, and we're actually able to identify transmission events of microbes from mother's milk to the baby's gut. And there's a very recent paper that we had, basically finding the same strains in mother's milk and a baby's gut, and those are overrepresented in pairs of mothers and babies. So this could be an interesting way to kind of understand how, where do we get the microbiome from?
It is. It is. So, and that for me is super, super interesting, because that's the very formative years where you're kind of really developing your microbiome, and maybe if you have antibiotics, it has a longer term impact, and it sort of just adds even more weight to the importance of breastfeeding, because it's both nutritional, but it also may be microbiome developing. That's just and one of the more interesting things for me for this study is not just look at how this early life microbiome is established, but also see how long does it stay. So we have some babies and kids that are 5 years old that we have for repeat visits, and we get samples from, so we can see whether they have the same strain still there. That's cool.
And we can also measure all kinds of outcomes, all kinds of disease, all kinds of health related clinical factors that we have for these kids. So we kind of see whether certain microbes, they got them from the mother, then how do they help the health long term of the baby.
That's awesome. Great. Well, we've covered the global microbiome, we've covered the baby microbiome, the mother's milk, we've covered all sorts. I think it would be really great, just as we kind of close to a wrap up, for you to let the listeners know where they can learn more about your work. How can they follow your research? Just that kind of thing would be really great, so that they can do some more reading.
Of course, yeah. So we have a lab website, blekhmanlab.org. You have a lot of information about our research there, links to all of our papers, contact information. If you're interested in contacting me or any of my lab members, specifically for the Human Microbiome Compendium, which is this big collection of 168,000 samples. This is all available online. But if you go to microbiome-based.org, you'll have all the information there in terms of, you can kind of have this high level overview of which countries we have, what taxa are found in each country. And if you want kind of a slightly deeper interaction with the data. You have there in the same website, you have links to our GitHub, which has all the code. We have an R package. So R is kind of something that a lot of people in the microbiome field are using to analyze the data. We have a package that allows you to even like download some of the data and analyze it yourself. So this is all available on that website. Yeah.
Great. Okay, Ran, thank you so much. It's been a real pleasure.
Thank you for having me.
Time's flown by.
Thank you for having me. This was wonderful.